Aind.
Yes, I mean,
When you are ready, we are ready.
yeah, okay. So this is the research I'm going to talk about. Uh, I think I have 25 to 30 minutes. So we're talking about the expression, purification, and characterization of SARS-CoV-2 of 9B and the study of its interaction with human H.S.P90 and cytosolic Tom70. So introduction. So in order to replicate efficiently, viruses they need precise interaction with the certain host proteins from the protein quality control, such as the chaperones and the H proteins. And also, in the other hand, several proteins they try to hijack the parts of the proteases system of the host, in which they use this system to upregulate or downregulate the physiological processes, or inhibit and activate certain cascades necessary for their own survivability. So here to introduce the SARS-CoV-2 genome, we have the open reading frames, the open reading frame and the structural protein embedded in the genome. Basically, what we're talking about here is the nucleocapsid protein, which contains the open reading frames 3A to 10. And our major focus here is the OF9B, which is a nucleocapsid protein from the genome of SARS-CoV-2. So, why are we studying the OF9B? It's the research has earlier been determined that OF9B can disrupt what we call the mitochondrial antiviral system. So the mitochondrial antiviral system is a very important innate immune response in which involves some proteins such as the HSP90, TOM70, the IRF3, and the TBK1. So this system, they are upregulated when there is antiviral attack, when there is viral attack, and so this system tends to produce the type 1 interference and eventually eliminating the RNA viruses through the innate immune response. So OF9B has been determined to disrupt these particular processes. So talking about the structure of 9B, into the short time into the proposal of this project, the structure, the carrying structure of the of 9B was determined, and it was determined to be an homodimeric form of homodimeric protein with amino acid residues of 97 amino acid residue, and also it the complex between OF9B and TOM70 was also determined, in which we see that OF9B occupies a pocket in the TOM70 structure, and this can be the mechanism in which OF9B uses to disrupt the mitochondrial antiviral system, because TOM70 is a major player in the antiviral system of the MAVS assembly. So the objectives of this study, we aim to clone, express, and purify OF9B, and to produce a pure form of the 97 amino acid protein, and to characterize the properties, the structural and biophysical properties of the protein. Also, we also aim to co-express the OF9B and the TOM70 complex, and to determine the stability and characterize the biophysical characteristics of the complex. Also, we aim to express and purify TOM70, HSP90, and the C-terminal HSP90 in order to study the role of 9B in HSP90 TOM70 interaction. So, what are the methods? Basically, we cloned and did the expression of proteins. Basically, all the proteins used in this study was cloned and expressed by me. And also purified by affinity chromatography and also gel filtration chromatography. Then also performed circular dichroism. SEC mouse is also used to analyze the the biophysical properties of the proteins and also to determine the protein-protein interaction. The tryptophan emission fluorescence was also used to determine the structural characterization of the protein. Differential scattering calorimetry was also used to elucidate the biophysical properties of the proteins in the study, and also pull down experiments was employed in order to see protein-protein interaction. So the results. So firstly, I did a pairwise alignment of the SARS-CoV-2 of 9b with the SARS-CoV of 9b, because the of 9b also exists in the initial strain of the SARS-CoV, which is SARS-CoV-1, which originated around 2005. So the structure of 9b and of 9b from SARS-CoV-2 and SARS-CoV-1, they have similarity complex of about 72.4%, which shows that the structures are similar, and they have some conserved sequences in the structures of both of 9b's. So the expression of 9b was cloned into the PET28 plus the site for the TEF protease and polyhistidine for the process of purification. So the molecular weight of 9b is 10.7 kilodalton, and with the E stack is 13.2 kilodalton. So firstly, we did the expression tests. So the expression test of 9b, we determined the expression of 9b in E. coli cells in different conditions and different environments. So we tried the 1 hour, the 2 hours, the 3 hours, and the 4 hours expression in 37 degrees and 20 degrees Celsius, and we determined that the best condition to express the of 9b is expression for 4 hours at 37 degrees Celsius. So now to purify the of 9b, which we through affinity chromatography, and we got a pure form of 9b here in the as analyzed by SDS page. Also, we further did the gel filtration chromatography of 9b, which also gives us a pure form of the of 9b of interest, and also to determine the purification, the purity of our form of our expressed protein. We perform dialysis using 3.0 kilodalton membrane, and also we analyze the SDS page in ratios, in the ratio 1:1, 1:2, 1:4, 1:8 of the of 9b and the loading buffer, and we signified that this protein is 97% pure. So because we have an histidine tag in the protein, so in order to keep the protein in its pure in its pure poly amino acid residue, so we tend to remove the histidine tag using the TEF protease, because we have the TEF protease sites on the cloned sequence. So here is the SDS page, indicating the two forms, which is the E tag form and the no tag form of 9b, and also to determine the effectiveness of our TEF proteases, we perform the Western blot of antihistidine Western blot analysis of the protein, and we found that the protein was detected and was not detected. The protein was not detected in Western blot analysis. So further, we performed the SEC mouse experiment to show to determine the molecular mass of 9b, and also we found out that the molecular mass is 22.8 kilodalton, and which indicates which is consistent with the structure of 9b, which was formed to which was determined to be a homodimeric structure. So this confirms that the protein is expressed as a homodimer. So in order to determine the secondary structure of 9b, we performed the CD experiment in which we was scanned from 205 to 260, and we found out that the minimum spectra was around 207 nanometer, which correspond with a protein with high abundance of random coils. So we found out that of 9B structure has high abundance of random coils in the structure, and in order to see if this random coil structure of 9B can also be altered and can be reshaped into the alpha helical structure, so we introduced the treatment of the of 9B with the TFE, the tetrafluoroethanol, with different concentration of tetrafluoroethanol, and we found out that this structure retains the shape of this random coil abundance structure. So further, we tend to co-express the of 9B and the TOM70 complex together. So the of 9B and TOM70 were co-expressing BL21 strain of the E. coli, and were heat shock transformed. And when they were heat shock transformed, we expressed we expressed and purified the protein. And so we found out that the protein was expressed as a complex. We have TOM70 and we have of 9B as after affinity chromatography. So, and after gel filtration chromatography, we found out that the highest peak here, the peak 3, also correspond with a complex formation of TOM70 and of 9B. So which will be referred to as the of 9B TOM70 complex. So in order to also study and differentiate, use TOM70 as a positive control to our analysis, we also tried to purified of TOM70 using affinity chromatography and also gel filtration chromatography, and perform the SECMAUS, the of 9B TOM70 complex, the analysis of 9B TOM70 complex, and determine the molecular mass of 9B TOM70, which corresponds to two corresponds to a monomer of 9B and a monomer of TOM70. So the complex is formed between a monomer of 9B and a monomer of TOM70. And here is a SEC chromatogram, which shows the shifts in the elution peak of 9B TOM70 complex, TOM70 and of 9B. So this shows that the of 9B TOM70 complex it's a complex formation between TOM70 and of 9B, and which is has a higher molecular weight than TOM70 alone and the of 9B. So I should sound a notice that the TOM70 used in this particular study is the cytosolic form of TOM70, which is from the 111 amino acid residue to the last amino acid residue of the TOM70 protein. Further, we did the tryptophan fluorescence spectrometry of the complex and also TOM70, and so we here the maximum wavelength, we excited tryptophan at 295 nanometers, and the maximum wavelength was determined. TOM70 of 9B TOM70 complex has a maximum wavelength of around 338, and TOM70 around 339. So this result shows and gives the fact that tryptophan is well embedded in the structure of the TOM70, and also the embedded structure of the structure of 9B TOM70 has not changed the overall structure of TOM70. So the tryptophan remain embedded in the structure of TOM70 of 9B. So this also gives the understanding that the of 9B complex formation with TOM70 did not disrupt the structure of TOM70 in any form. So this is circular glycogen spectra of the of 9B TOM70 complex and compare with the TOM70. So here we found out that these two also the minimum signal is at 222 and 208 nanometers, which indicates this structure are, they are abundant in the alpha helical conformation. So this also shows that when of9b forms a complex with Tom70, it's in the alpha helical conformation. Likewise, the Tom70 complex, the Tom70 protein alone. So here we did the CD thermal unfolding, in which this CD spectra was read from 20 degrees Celsius to 90 degrees Celsius, and used to compare the Tom70 and the of9b Tom70 complex. Here we found out that the melting temperature of the complex is around 59 degrees Celsius, while the melting temperature of Tom70 is 49 degrees Celsius. This shows clearly that the complex between of9b and Tom70, the formation of of9b Tom70 complex has stabilized the structure of of9b Tom70, which makes the structure more stable than the more heat stable than the Tom70 alone. Here we determine the differential scanning calorimetry of 9b Tom70 complex, which also was in accordance. It agrees with the result from the CD experiment. So this also shows the melting temperature at 49 and 58 degrees Celsius respectively. So we have the of9b Tom70 complex, we have the Tom70, and we can see that the melting temperature is more increased in the complex than in the Tom70 alone. And also we can find here that we have some we have a minor melting temperature, which can be as a result of the Tom70 structure, which contains some dimeric form which can be seen through the Sekmaus experiment. So to determine the protein-protein interaction, we start by using Tom70 as a bait, and of9b to attack to see the interaction between of9b and Tom70. And we found out that after the incubation of Tom70 and of9b, there is a minimal there's a complex which involves a minute, a small concentration of 9b. This results, excuse me. This result is particularly important because it confirms that when Tom70 and of9b are physically mixed together, this complex is not properly formed, because of9b it occurs as a homodimer, while Tom70 is also is a monomeric unit. So the homodimeric formation of 9b is not able to form a proper complex with Tom70, because we determined that the complex is properly formed between a monomer of Tom70 and a monomer of 9b. So here, in order to continue our protein-protein interaction study, I purified the full-length human HSP90 using affinity chromatography, and likewise using gel filtration chromatography. And also, we performed the TEF protease removal of the polyhistidine tag in the HSP90 structure, and confirmed this using Western blot analysis. So this is the pull-down experiment of Tom70 and HSP90. So Tom70 alone and HSP90 was determined to form a complex with each other, which is has been experimentally determined. We use Tom 70 as a base, which contains the histidine tag. The HSP90 without the histidine tag is used to to study the interaction between Tom 70 and HSP90. So after the incubation and so many wash and the incubation with HSP90 and 3 CV of washing, the proteins were eluted as they were eluted together as a complex by imidazole. So this experiment was repeated using the of9B and Tom 70 complex with HSP90. So the of9B and Tom 70 complex, refused. It did not, form the interaction with HSP90 because after we incubated HSP90, after the wash of after we incubated the Tom 70 of 9B complex and we incubated it with HSP90 and wash, we discovered that HSP90 was washed through the column and the elution was the elution contains just of 9B and Tom 70 and of 9B complex, which shows that HSP90 did not interact with the of 9B Tom 70 complex. So this result is telling that of 9B Tom 70, the presence of 9B in Tom 70 is preventing or not allowing the interaction between Tom 70 and HSP90. Further, we did a SEC analysis, the SEC experiments to also show the interaction between the complex of 9B Tom 70 with HSP90 and the two proteins were incubated and injected into the SEC column, the GF column, analyzed using the SEC mouse machine. So we found out that there's two distinctive peaks, which after SEC SDS page analysis shows that it's a distinctive peak of HSP90 and a distinctive peak of Tom 70 of 9B and Tom 70 complex. So which also which agrees with the previous experiment that HSP90 did not form an interaction between of 9B Tom 70 with of 9B Tom 70 complex. So this here also shows that we treated the HSP90 with ATP and ADP because the structure of HSP90, there's a conformational change when HSP90 is bound to ATP and ADP. So we treated HSP90 with ATP and ADP and also tried to see the effects in the interaction with of 9B Tom 70 and this similar peak, which are distinctive of HSP90 and of 9B Tom 70 complex, was realized. So further, in order to ascertain this interaction and non-interaction of HSP90 and Tom 70 of 9B Tom 70 complex, we also purified the C terminal of HSP90 because the C terminal of HSP90 is the region of the protein which forms the interaction with Tom 70. So in order to rule out other form of interaction in the rest of the structure of HSP90, expressed and purified the C terminal HSP90 through affinity chromatography and gel filtration chromatography, and use this protein also to study the interaction between Tom 70 and C terminal HSP90. So here we find that when we perform the SEC analysis of the incubation of C terminal HSP90 and Tom 70, so we found out that at this peak there is a complex formation between Tom 70 and C terminal HSP90. Unlike when we incorporated and performed the second analysis of C-terminal heterospinality and the of 9b Tom 70 complex, so there was also distinctive two peaks in, and the first peak correspond with the of 9b Tom 70 complex, while the second peak correspond with the C-terminal heterospinality. So this also was showing the fact that of 9b Tom 70 complex did not bind with C-terminal HSP90. Conclusions. So conclude that the recombinant SARS-CoV-2 of 9b is a soluble protein which can be expressed in E. coli system to obtain a pure form. Also, the SARS-CoV-2 of 9b is a homodimeric protein with molecular weight of about 22 kilodalton, 22.3 to be precise. And this protein has a high portion of random coils in a secondary structure. Also, the SARS-CoV-2 of 9b is mainly in a random coil conformation. The co-expression of 9b and Tom 70 produce a stable of 9b Tom 70 complex. And also the complex is more stable than Tom 70, is more stable than Tom 70 alone. And the C HSP90 and the HSP90 full length binds to Tom 70 alone and not to of 9b Tom 70 complex. So these are my references. I'd like to thank my supervisor, Professor Carlos Ramos, for his wonderful support and his fatherly advice and affection in the lab and outside the lab. Also, I give so much thanks to Glaucia. She's very, very helpful, throughout my research, and also our technician, Dr. Annelise. She's very, 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 very supportive and helpful. All the committee members for being here today, I really, really thank you. I appreciate it. My colleagues in the labs and my friends and family that are present to grace this qualification exercise. Thank you very much.
Very good, Ayinde. Now we have the arguation. We are going to start with Leandro. Now we can start, Leandro.
Okay, yeah.
Nice, fine. I think you are the expert because my contribution here is going to be very...
Okay, so thank you very much. Thank you for the post-graduation of Chemistry Institute to accept my name. Thank you, Ayinde, to invite me, and also Carlos.
You're welcome.
I am very, very familiar with your work because I talk very, very much with Carlos, and and I know very, very much of your study. I would like to start by you making some comments for me
Okay.
about the stability of the protein, the ORF9b, together with Tom 70. You show here the DSC.
Yes.
Of the complexes.
Yes.
Yeah, I think it's this one. I think it's 27, if I'm not wrong.
Yeah, the DSC. Yes.
And you make the Tom 70 alone, but you did not measure the ORF9b alone.
Okay. So I measured the of 9b alone. It's because it's a random coil protein. So the of 9b alone, it's did not melt at all at 20 degrees to 90 degrees.
Okay, so
The
there is
did not melt.
No phase transition there.
Yes, there's no transition in the of 9b alone protein.
Okay.
Yes.
But here you see that there is like a shoulder around the 52. Yeah,
yes.
There. Yeah. Do you think that this could be the unfolding of ORF9b? Because when it interacts with Tom 70, it may, for example, gain a little bit of secondary structure.
Yes, it is very quite correct that when OF9B forms the structure with Tom70, there's a particular region between the binding site of the OF9B with the Tom70, which later assume the alpha helical conformation. So because
like inside that pocket that you show.
Yeah, in the side inside the pocket of the Tom70, there's the gain of alpha helix conformation in the OF9B structure.
I see.
Yes.
And in the same direction, in the same argument, too, then you can comment a little bit about the interaction with the AGSP90.
Okay.
I think it's this one.
Yeah.
Because here I did not quite understand the difference between the black and the red curve here in this.
Okay. So this here is the black is when HSP90 was treated with ATP, and the red is when HSP90 was treated with the ADP. So we also use this, to see the interaction between OF9B Tom70 also. It's complex.
But they are quite similar.
Yes, it's similar because the HSP90 is the same, the OF9B Tom70 complex is the same, but what we try to see is that what happens when we treat the HSP90 with ATP or ADP.
Okay.
So the ADP the protein is in micromolar concentration, while the ATP and ADP they are in the molar concentration is very, 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 very small, very, very minute regarding the protein. Yeah. So we found out that even in the presence of ATP and also in the presence of ADP, there's no bond formation there's no interaction between the OF9B complex and the HSP90.
I see.
Yes.
And talking a little bit again about the X-ray structure of 9B, I think it's figure 4 or figure 5 of your thesis on page 15. You have there the homodimer structure, but there is a lot of beta sheet structure on that figure.
Yes. So because the structural determination of 9B, it's always referred to as a beta sheet structure. Okay. So
but you don't see this kind of structure in solution according to your CD experiments in this slide 21.
Yes, our CD experiment shows that even in this beta sheet structure, we can find that the abundance of the structure is also random course. In a random course, to form the abundance of the sequence of the protein. And when we talk about CD structural determination, it's always quite distinct from the structural determination when you talk about X-ray crystallography and maybe chirom and legs. So the CD, it sees the global structure of the protein. So it tends to give us the most abundant secondary conformation of the protein of interest.
Okay, and when you see this kind of structure, like in figure 5, and you see the pocket inside the Tom70 in figure 6, for example, it's just a small part of the protein that interacts with this one.
Yes.
Do you have an idea? Is the N-terminal, C-terminal? What region is that?
So the region of the Tom70.
No, of 9B.
Of 9B. So it's of 9B is a monomer, okay, and the region between the 26th amino acid to the 54 amino acid thereabouts forms this particular interaction with Tom70.
54 residues, more or less.
Yeah, more or less, yes.
Or more or less half of the protein.
Half
because
of the
it has-
protein.
okay,
Yes,
okay.
it's more or less the half of the protein is forming the interaction with Tom-70.
But you have to make like you have to unassemble this dimer to make this kind of interaction. Or do you believe that the dimer conformation of ORF9b is kept during the Tom-70 interaction?
No, the dimer form of the of 9b it's not existent in the complex. So that is why it is very difficult to physically form the interaction. That's why we decided to go through the process of co-expressing the protein together, so that the complex will be in its right conformation of one monomer of 9b and one monomer of Tom-70.
Okay. So okay, I see.
So also this was confirmed recently through some paper, which also shows that of 9b homodimer does not, is not able to form the interaction with Tom-70 unless it is in the monomer conformation.
Okay.
Yes.
Okay. So somehow Tom-70 makes the protein to melt, let's say, this dimer.
Yes, and it form the bond with the monomer.
Okay. And then you have the interaction with the monomer. It's quite interesting. This it's not so common, this kind of behavior, no?
Yes. So
I have another question regarding the. Okay, these experiments, do you have an idea? Of course, you made everything in vitro. I mean,
yes.
And in vivo, do you think that this interaction also happens in vivo? The interaction of ORF9b with Tom-70, for example.
Yes, because there was a study in which when they extract the cells from the lungs of patients that are infected with SARS-CoV-2. So this shows that they found out that there are a lot of expressed form of 9b and Tom-70, and this was also very, very implicated in the antiviral mechanism, especially the formation of interferons. Tom-70 is very important in interferon formation, and also a particular study shows that when Tom-70 is binding with OF9b, they used a human cell model to show that when Tom-70 is binding with OF9b, then there's the blockage of the formation of interferons, which enhance the antiviral, the viral, the infection of the virus. So I think I can say that the formation also occurs in cell. In vivo.
Okay, very, very nice.
Yes.
And you make also, of course, several biophysical experiments, but you did not mention anything about the structure itself. What kind of measurements regarding the structure of the protein and the complexes? For example, do you have any plan? Oh, of course, before I make this question, I have to ask you, what are your plans for the future? I mean, are you keeping to work with that system? Are you changing a little bit of systems?
Okay, so the next experiment is to see if the complex, because we already know that the interaction is the interaction of 9b and Tom-70 is precluding the formation of a bond with HSP90. So this also can be further studied to see the specific manner in which this interaction is being precluded. And most specifically using biophysical techniques also.
Okay, but are you continuous with this work, or are you doing the PhD here, or are you coming back to your home? What are your plans in that regard, actually?
Yeah, I am planning to continue PhD in protein studies also.
Okay, but not specifically with that system.
Yeah,
Because,
not,
for example, you can make some. I work very much with small angle X-ray scattering,
okay.
and this is a quite nice
SACs.
system. Yeah, SACs. Yeah, it's a quite a nice system to measure because you have monomers, dimers, complexes. Yeah, I mean there is a lot of fun to work with that system. So if you'd like to make some kind of small angle X-ray scattering experiments, and here
Okay.
if you need.
Okay. Yeah. Thank you very much.
I think I'm very, very satisfied with your presentation, your work. I know very much your work because I've talked to Carlos very much about this. And congratulations, I'm very, very satisfied. Thank you very much.
Thank you very much, you're welcome.
Bispo, estou bem satisfeito. Pode prosseguir.
Legal, Leandro, obrigado. Okay, now Eneida.
Thank you, Bispo. So I would like to thank AYINDE and Dr. Carlos Ramos for inviting me. It was quite an adventure because I'm not that near to protein studies anymore. It has, since it was a while then from when I started working, or when I worked with protein structure, but it was very nice. I enjoyed reading this. And I have a specific point that I would like to ask you. First
Okay.
of all, when did you start your master's?
I commenced in March 2020.
2020.
Yes.
So the pandemic got you?
Yes, yes.
And could you stay at the lab? Could you conduct the experiments? I mean, the expression and the purification. Could you do that there, or did you have to stay at home?
I stayed at home throughout last year. So although I have some days I go to the lab last year, which I got permission from the institute, but basically this work was commenced in January 2021.
I see.
The lab work commenced specifically in 2021 about this particular project. Yes.
And did you have any previous experience with protein expression purification?
No.
So you had to learn everything.
I learned everything through my lab members and my professor. Very very good. Yes.
Okay. So you were a survivor from COVID pandemic.
Yes.
Okay. Okay. When you start studying this, when you start with this project, the idea was to solve the structure of or 9B, or because I think it happens during this time that they discover the structure. Is that?
Yes. So the idea, the basic idea was to, study the structure of 9B. So as we go into the projects, as various researchers from everywhere, start to start to have so many answers about of 9B. So we tend to improve the study and the plan of the particular projects.
I see.
Yes.
Okay. Well, one point that it didn't, I could not understand is about the dimer and the monomer, and you have purified the dimer of 9B dimer.
Yes.
And when it binds to ton 17,
yes,
it assumes an helical conformation, and it becomes a monomer. I didn't understand I didn't understand well. How did the transitions from the dimer to the monomer occur?
I think this is one of the cellular organization of the proteins, because this transition or this interaction never occur when we physically mix the two proteins. It never occurred. So it occurs when these proteins were allowed to express together. So when the proteins are expressed together, then it was expressed in the conformation of a monomer, in which the of 9b exists as a monomer in the conformation of Tom 70 complex.
I didn't get this idea by reading your text. Maybe I'm not from the area, but I didn't get this idea that you only get that when you co-express the two proteins. So maybe you can make sure that this is on your test. This is quite clear, you know.
Okay.
Maybe it was just my question. Let me see my questions here. That publication of yours, you have a publication from last year, yeah?
Yes.
I could not take the whole PDF. What was that about? Because it's not about your results, yeah?
Yeah, it was about it was a review article.
A review, okay.
Yeah, a review article describing the role of HSP7, HSP90 in viral infection, and specifically for SARS-CoV-2.
Okay, so that's what I thought. It was a review article, so
yes,
you were putting the ideas together, but you didn't publish your results.
No, not yet.
Okay.
Yes.
Okay, let's see here. I would like to know more about the SEC mouse technique. What's the limit of detection of this technique that you used to set the molar weight of your protein?
So the technique is basically the size exclusion chromatography, which is coupled with a multi-angle light scattering device. So the multi-angle light scattering device, it scatters the protein, the light on the protein, and measures the scattering at different angles. So we have the 90 degrees, the 180 degrees, and our machine laboratory specifically, records the scattering at 90 degrees. So this detector, they contain a refractive index detector, also a Stokes radius detector, and these detectors, they use the concentration of the protein to determine the molecular weight, the molecular mass of the particular protein in which it's being studied. So when the gel filtration chromatography, when it separates the protein of interest, which is also detected by the UV, now each protein separated undergoes this scattering, and we determine the molecular mass of each protein using the software, because the device has a software referred to as the Astra software. So we determine the structure, the overall molecular mass of the protein using the software, the Astra software.
I see. But my question is, if you have an idea of the sensitivity of the technique, I mean, for what is the range of molecular size that it is able to detect, to separate? As it is for protein, I imagine
it
it's
is
for
protein.
large.
Yes, it's for protein. I am not sure the range or the limits. I'm not sure about the range or the limits. But it measures as big as dimers, tetramers, and trimers.
Do you know, Leandro?
I think
Just a curiosity.
I think that the small limit should be something around 5 to 10 kilodaltons, and the larger size is something around a few megadaltons, like 1, 2, or 3 megadaltons, more or less this range.
Okay.
Okay.
Thank you.
Well, one other question is about the DSC experiments.
Okay.
And I would like to know, I mean, you had a change from a 10 degrees change in the melting temperature.
Yes.
And but as I'm not in the area, I would like to know if the changes in enthalpy says it's a strong interaction or not. What can you tell me about the size, the dimension of the enthalpy change that you got? What it can say about the interaction between these two proteins?
So
Is it a strong one, a weak one?
yeah, so the enthalpy, it shows basically the concentration of the proteins. So the enthalpy difference, gives us the temperature, the concentration. It's related to the temperature of the protein in which we injected in the DSC machine. But the the melting temperature, because the TM, the melting temperature, gives us the stability of the protein at the presence of heat, increase the percentage of heat. So in this particular experiment now, so we found out that the of9 bitum-70 complex has a very high melting temperature compared to bitum-70, which signifies the stability of the of9 bitum-70 complex than the bitum-70.
Yeah, I understand the changes in temperature. I agree. It's almost 10 degrees, so it must be an important interaction.
Yes.
But I cannot translate this in enthalpy numbers because I'm not used to these enthalpy numbers.
So enthalpy numbers is just the molarity is just dependent, because if you see the unit is in the kilocal per mole per degree Celsius. So it's
but I insist, the size of this change can tell you about the interaction, how strong is in the interaction.
Yes.
I don't know if it's a strong one or a weak one. And if you don't know now, please just think about that and try to know if this enthalpy describes a phenomena that is I was thinking. Okay. The of like a titration.